three neat kind of topic areas related to gene expression. And we're going to look at all three very, very quickly. So the first thing I would like to talk about is gene mutations. Okay, so gene mutation. Where does this come up again? Gene mutation. Can you remember? In year 12 genetic diversity. So you do about different types of mutation. There are 5 different types of mutation, and I am not going to go through all of them today. Okay, I'm going to go through some of them first of all. Okay, so you'll know about substitution. Substitution. It's where one base is replaced by another. So one base is swapped for another. Is that pretty catastrophic for somebody? No, why not? Okay, it doesn't cause a frame shift, and actually most AAs, they have multiple triplet codes. So you don't always get a change. And it's known as a degenerate code. Well done, degenerate. Very good. So it's known as a degenerate code. So, for example, let's say we got CAC or CAA, okay, and we get a deletion, okay, and then you get that as CAT, but it is the CA and the CA that determines the amino acid lysine, for example, okay. So if it's on the 3rd codon, it usually doesn't have an effect. I just made that up, by the way. Do you need to know the triplet? It's a bit quick. Triplet codes for the amino acids? No, you do not. And you would be given that evidence in a question. Good, right. So deletion. So that is where a base is removed. Okay. So this does cause what we call a frame shift. So basically downstream of the thing, the whole code moves, and therefore many amino acids can change, causing proteins to be non-functional. So others that will change the amino acid, one is addition and duplication. So addition where they add, or duplication where you get copying the same basis. So that does cause frame shifts, so they're pretty bad. Other ones though, so we've already mentioned substitution plus inversion don't really have a massive effect. So sometimes they don't change the sequence either. Good. So that's all I'm going to do about gene mutations. So actually, we haven't said how they're performed. It's an error in the DNA replication or due to mutagenic agents. And obviously a key mutagenic agent, obviously, is the radioactive spider, as we all know. It is, look, it is April the 1st. Anyway, yeah. Okay. Oh, I'm the fool. Damn it. Do you know, I was going to ask Tom Cruise to come today and do it, but I'd say, thank God I didn't know. Right, private joke with my students. Let's talk about stem cells. I'm going to talk about stem cells now because I like stem cells. And I don't care if you got stem cells. Okay, so
Stem cells, they are undifferentiated cell types, okay? They become specialized. So they're unspecialized cells that become specialized. There are different types of cells. So in the embryo, for example, you can get, well, what can you get in the embryo? In the first few cell divisions. So first few cell divisions, they are what we call totipotent. So that's totipotent. So they can form into any type of cell, specialize into any type of cell. Okay, so they specialize into any type of cell. Oh, this might be worthwhile knowing, it's only in mammals. I don't think that would be useful. Okay. Then we've got multipotent. So they can differentiate into a few different types. So e.g. blood cells. And then last one, we've got unipotent. It's interesting unipotent because they mentioned specifically a type of unipotent stem cell that I'm going to discuss in a minute. So cardiomyocytes. So only one type. So for example, cardiomyocytes. Where would you have cardiomyocytes? What does it sound like? Heart muscle cells. Good. So heart muscle cell. Okay, so they can be replaced. Also epidermal skin cells. Okay. Now, obviously, they want you to know about stem cell therapy, about genes being expressed. Okay, so stem cell therapy. So therapy, they can be used for human illnesses, okay, replace damaged cells. So some type exists, so we already can, for leukemia, we can replace red blood cells, RBCs. You can take it from the bone marrow, so stem cells from the bone marrow. I'll be honest, I think that they would probably give you lots of this information, to be honest. So they might give you an example and they might ask you to discuss. Huge benefits, save lives, reduce suffering. Where do you get them from? Okay, so they're from two places. They can be embryonic. Obviously, there is a ethical issue of using embryos. Where do they get embryos from? IVF, yeah. So they would have been thrown away anyway, but IVF. And then obviously adult stem cells, okay? So adult stem cells that only produce a small number of different types. Let's go down to the therapy, and I want to talk about IPS. What's IPS, please? Yeah, induced pluripotent stem cells. So you can reprogram adult body cells. Okay. To make them behave like embryonic stem cells. You can actually use what we call transcription factors. So transcription factors, I'll talk about what those are. Causes adult body cells to express genes. So to express certain genes. How can you introduce them into the cell? Introduced via a viral carrier, by a specially modified virus. So it gets them into the cell. Right, I think the big one, though, is regulation of gene expression. So we'll talk about regulation next. So switching genes on and off. So transcription factors. So obviously your body, you don't want to be producing lots of proteins all the time. So your genes have to be switched on and switched off. And that is through thousands and thousands of different types of transcription factors that control the target gene. And some of those you need to know about. One in particular is estrogen, and also the other one, so that would promote transcription, estrogen. The other one is about inhibiting it, which is the use of RNA interference, or siRNA and miRNA. Okay, so let's talk about initiating transcription. So there is a synoptic element here, although if it's not in paper 3, you might not have it, so I'm not going to mention. Obviously, the synoptic element here is with protein synthesis, but protein synthesis is in paper 1. This is in paper 2. So if there's a question about gene expression, they'll probably link it to protein synthesis in paper 3, but I don't know whether they'll do that. Shall I go on film now and say that I think there might be an essay on using gene technology? Don't know. Maybe not. Mrs. Miller will call me. Right, so, because you have to revise everything, of course you do. Oh, I've cut. Okay. Okay, so it's quite complicated, to say the least. Okay, so let's say about turning genes on. Obviously, outside, here's a nucleus. Okay, so here's a nucleus, and inside I've got my DNA, double helix, kind of. Okay, there's my DNA. Right, so out here, okay, is transcription factor, and here is some estrogen. Okay, so here's estrogen. So basically that can't travel into there to start transcription. Okay, so it has to bind. There we go. So you can see there's a complementary shape, and the estrogen can bind with the transcription factor. Transcription factor. Okay, so it binds and it changes the shape of thing. There you go, the transcription factor. So it makes an estrogen to estrogen receptor complex. And by binding, it then can be soluble and it can move through. Okay, and it moves through into the nucleus and it binds to a specific site. So it moves into the nucleus and binds to a specific site on the DNA. Very much about specificity. And it activates RNA polymerase. Okay. So it activates transcription. Yeah. siRNA. So let's turn off the gene. Okay, I'm going to talk about it's known as siRNA. It can be used to stop genes being expressed. Okay, what is it? It's a small double-stranded piece of RNA. It's small double-stranded. Oh, I'm running out of time. Okay, so what happens is the mRNA, okay, once it's been transcribed, it moves towards the ribosomes. When it's moving towards ribosomes, something happens to it. So siRNA has a base sequence that's complementary. Try to use those words. Base sequence that is complementary. And therefore it binds onto the mRNA. Okay. It binds to mRNA. And then what happens is that there's lots of enzymes, something called RISC, but in AQA you don't need to know about RISC. You just need to know that it then cuts the mRNA fragments, so it can no longer be translated. So it's used a lot in biotechnology to switch genes off to see the effect of switching a gene off would have. So I'll put this up. So epigenetics is really interesting. It's about it's very simple actually. So it's a new science and it is quite amazing. So basically you are born with all the DNA that you're ever going to have. However, some of that DNA is expressed, some of it isn't. Okay, and therefore it's about gene expression. So you obviously inherit certain what we call markers from your parents, okay, and most of them are removed between generations, but some escape it. So expression of some genes of offspring can be affected by environmental change. So that's the big thing. So some gene expression is controlled by the environment. Which is really interesting. Because it kind of is not very Darwinian, this. So it's saying what you do in your life could affect your offspring. And also during your life, how you live your life can affect which genes are expressed and which aren't. Okay, so we're going to talk about the ways that DNA is switched off and on. Okay,
So DNA can be switched off by a process of methylation. So increased methylation. So what is that in a nutshell? Basically, it adds a methyl group to the DNA, two bases in the DNA, okay, and it changes the DNA structure. So it attaches methyl group to bases, C and G. I think it's C and G, might be wrong. No, I'm right. Causes the DNA to coil tightly around the histones. Coil tightly. So basically no transcription can occur. Switching genes back on. So gene switch back on is acetylation. Gene switch on. Okay, so you've got histones that are proteins, okay, and then acetylation occur. So the addition of acetyl group makes the chromatin is less condensed. So added acetyl group leads to chromatin less condensed. So therefore, transcription can occur. Obviously, if you've got less acetylation and more methylation, the opposite occurs. So you can understand the role of genes. You could maybe understand the development of disease like cancer. So things like tumor suppressor genes. I haven't mentioned cancer, which is a real shame, actually. So I'm not going to have time for cancer today. It's very applied, this bit, so I'm not going to go through that. So you'd expect to see lots of information. So there we go. I would probably say plus cancer. So don't forget about cancer, which is oncogenes, proto-oncogenes, and tumor suppressor genes. There we go. Okay, thank you very much. Thanks for coming. And